Well, good afternoon, everybody. My name is Wolf Heyer. I have the honor and privilege to introduce today's star speaker, Dr. Bernard DeMassey. Want to welcome you to this special occasion. Bernard is a major contributor to our understanding of the mechanisms of meiotic recombination. He has a long scientific journey through many model systems, making many contributions. He obtained his PhD at the Université Paul Sabatier in Toulouse, working on E. coli DNA replication, and he discovered the TUS termination, the TER element that forms the termination system with TUS factor to unilaterally halt the replicative helicase in E. coli. It's a major discovery that's taught in every good molecular biology class, as some of the students from my class will recognize. He then went to do postdoctoral research with Bob Weisberg, initially at the National Institutes of Health and then later at Columbia University, where he did some of the earliest work on structure-selective endonucleases. He used the gene 3T7 enzyme as a model, but it really is a class of nucleases that cleave structured DNA into linear products. From there, he started to work on yeast meiosis and really developed that interest in meiosis to define not only the cis-acting elements, but also the trans-acting factor that govern initiation of meiotic recombination. And he made incredibly important contributions in the early days when it was just barely established that double-strand breaks is what induces meiotic recombination. And then in 1998, he moved his program to Montpellier at the Institute of Human Genetics and started to work on mice. And in a very systematic fashion, decided that he had to go back to what is a good cis-acting site to study, and he identified a meiotic recombination hotspot and characterized that. And then what are the trans-acting factors governing that cis-acting site? So a very classic approach, and that approach netted the identification of PRDM9 histomethyltransferase as a key determinant of initiation of meiotic recombination in eukaryotes. That was a stunning breakthrough for the field, and he has really made a very, very significant contributions in that. And there were other laboratories coming, homing in on PRDM9 from completely independent approaches. He has continued making really major strides. He has identified the partner of the SPO11 topo6 type enzyme that is the enzyme that makes the double-strand break for meiotic recombination, and they are continuing to work on this mechanism, what really governs meiotic recombination. Not surprisingly, Bernard has won many accolades for his work. He won the CERNAS medal, he won the Jules Martin Prize of the Académie des Sciences in France, and I'm sure there are many more prizes on the horizon for his fundamentally important and beautifully executed work. I'm so happy he has come to visit us. We had a wonderful time with him, and I can't wait to hear his seminar. Bernard, welcome to UC Davis.
Thank you. Thank you very much, Wolf, for the invitation. I'm extremely honored and pleased to be here and to have spent some time to communicate to you our main interest and a few results, some published and some unpublished. And for me, it's very exciting, always very stimulating to come to UC Davis and meet my friends and colleagues and to learn about the great science that's going on here on this campus. So I'm going to really present you the recent work about the topic in my lab, which is really meiotic recombination. And as the title says, there is a mechanical aspect, which is control of chromosome segregation, and there are evolutionary implications. And you will see that during my talk, how these are links. I'd like to start with really pointing out this early fantastic era of genetics at the beginning of the 20th century. And if you have not, I think it's always great to go back and read some of these papers from Morgan, Muller, and others. And this is really the roots of the events that we are still currently working and trying to understand, and which came about with the chromosome theory of genes, right? And the first genetic map that is from the Sturtevant in 1913, one of the first crossover model also, and together with the cytology, also very exciting at this time, in particular from Jensen, who set out the hypothesis for the chiasma connection between the chromosome during meiosis. And the link between all these two is what we are trying to work out the molecular details of. Genetic map, genetic distance, of course, raised a lot of questions about recombination. And as Hermann Müller writes here, based on the chiasma type theory, the frequency of recombination must be, to a certain extent at least, an index of the distance. And this index of distance is what have been really one of the challenge in the field. So these chiasma are formed during meiotic prophase, and so they lead to a crossover, which determines the genetic map. And what this exchange allows to do is to connect, as you can see here, the two parental chromosomes. And this connection is a mechanical link, which is essential for the segregation, and it's just essential for fertility. So it's a key essential step of sexual reproduction. And of course, in addition to that, these exchanges increase genetic diversity. So at the same time, there is a mechanical role with evolutionary implication, which have still remain a lot to be done to be understood. So meiotic recombination is taking place during this prophase. And now just a little bit more information is that we know in many species that the exchanges by homologous recombination it's a programmed pathway that initiates with formation of breaks and rejoining these breaks to complete the exchange. Of course, you have to exchange 2 DNA helixes, so you have to cut somehow and religate 4 strands, and this is taking place in a highly coordinated and regulated pathways by involving many proteins. So this prophase can be followed by a various cytological marker. I'm not going to describe the cytology because we're going to go into molecular details, but if you are familiar with the double-strand break response, also you may know the phosphorylated form H2AX, which is called gamma H2AX, which essentially covers almost the entire chromatin in the nucleus at the beginning of this meiotic prophase, which is an indication of the formation of many breaks, and we have an estimation of this number in mammals, and it's several hundreds of them which are formed. And the question is, how are these breaks formed and how they are generated? It's a very challenge for genomic stability. It's actually crazy, you would think, to introduce several hundred breaks at the time where you're going to transmit your chromosome to the next generation. However, this is taking place. And these are the various topics that we are interested in my lab, together with implication on evolution. And today's talk is going to be focused on the break formation, and essentially these double-strand break, how they are formed and where they occur in the genome. Drawn here is a very schematic view of chromosome organization. I will get back to that later in the talk. So how do the breaks are formed? One of the first direct molecular link came from studies in yeast, And in 97, a paper that I contributed to in Alain Nicolas' lab in collaboration with Patrick Forter led us to identify that SPO11, a yeast mutant defective for double-strand break formation, is actually orthologous to a catalytic subunit of a topoisomerase family called topo6. And the catalytic subunit is called topo6A. At the same time, Scott Kinney in Nancy Kleckner lab detected SPO11 as covalently attached to the ends of meiotic DNA extracted from meiotic cells. So that came to make a very strong prediction that SPO11 would be the catalytic activity for inducing break formation in a mechanism that could be in part similar to cleavage induced by DNA type 2 topoisomerase. So a lot of biochemical studies on topoisomerase now are going to be important to present you the question that were raised. The topo6, which is an archaebacterial, first characterized in Archaebacteria, is as a catalytic subunit, topo6A. Here it's the crystal structure had been determined, and the two monomers introduce a coordinated single-strand NICs to make a double-strand break on the DNA. But the important information that I would like to highlight from the topo6 biochemistry is that this catalytic subunit requires another subunit, a B subunit for topoisomerase activity. Also to present how this topoisomerase like topo6 reaction works is first the enzyme is an heterotetramer, 2A subunit, 2B subunit. The B subunit has a domain called GHKL, which is an ATP binding, ATP hydrolysis domain found in a variety of proteins. And it has another important domain called transducer. I will go illustrate what these two domains are in the complex that you can see here. So this is the A2, 2A subunit here at the bottom. That's the DNA binding, the catalytic core of the complex. And the A subunit, just remind you, is the one that is orthologous to SPO11. The B subunit, these two domains are indicated here. So the GHKL domain has an important property, which is to dimerize upon ATP binding. The transducer domain has an important property, which is to interact and to stabilize the complex with interaction with the N-terminal region of the A subunit. So topoisomerase, which again biochemistry had been studied in many details, do cycle of cleavage and ligation to regulate DNA topology. And I'm not going to go, of course, into the details, but schematically you can see here that the complex has the ability to bind DNA to induce a double-strand break. For topology, of course, regulation, a second DNA duplex is trapped. What's important to notice is that the monomer interface and the dimerization steps are key in this, for this DNA cleavage and religation, in particular through the dimerization in the GHKL domain. So the question was, since SPO11 had the homology with the A subunit, how could SPO11 work alone? Is it possible without a B subunit, or is there a B subunit that also acts during meiosis for the breaks that are catalyzed by SPO11? And this question had been raised since 1997, and we got the answer just last year. So the answer came by collaboration with a group working on Arabidopsis who identified a gene which is required for meiotic double-strand break formation. And Mathilde Grelon, who kindly gave us the sequence of this plant gene, allowed us to search for orthologs in other non-plant species. And thanks to a great collaboration with Henri-Marc Bourbon, who is a friend and colleague I had collaborated for other projects before. He has a fantastic expertise in actually identifying remote, weak homology amongst proteins. And indeed, he was able to identify, you see these very weak regions of homology between the plant protein and here a mouse protein. Of course, you're not going to be convinced by these small boxes. So that's one among many analyses that Henri Marc did. But that led, in fact, to the identification of the whole series of proteins in plants and animals, which were predicted to be orthologous. This alignment allowed to further searches and to actually search by running structural prediction algorithm such as HHPRED, which immediately led us to identify that this family of protein revealed a very strong and significant similarity with the B subunit of type 2 DNA topoisomerase. So we were very excited that actually we had in hand a potential B subunit in mammals, in plants, and potentially involved in the interaction with SPO11. The sequence of this plant and animals B subunit, so we called this protein top6B-like as the first approximation, had indeed a structural domain similar to the top6B from archaebacteria I mentioned to you before, the GHKL and the transducer domain. More precisely, when you looked at the alignment and secondary structure prediction, essentially the GHKL domain is made of sandwiches of beta sheets and then loops arranged with alpha helixes and some small motifs which are important for ATP binding. So the overall structure was predicted to be conserved in plant and animals proteins. In terms of the primary amino acid sequence conservation, it is detectable. It's very weak. That's why probably it had not been picked up in the preceding 20 years or so. But we could detect some conserved motif with quite some divergence. So that's why this time we call it B-like, with, as I will come back to this, the questions that are raised by this. We were able to run a structural prediction model, being aware of this, that are just prediction based on the alignments. So that's of course intrinsic limitation, but it's possible to actually fold and organize the mouse top6B-like in a structure that is highly similar to the archaebacteria protein and without distorting the backbone in a bad manner, and that can be assayed with quality scores. So this is a GHKL domain, appears to be potentially structurally conserved in this family of proteins. And the other important domain is a transducer, which is actually essentially a very long alpha helix. This is archaebacterial with a turn and another helix. And this structure is also predicted to be found in this mouse and also the plant proteins. Amino acid conservation is poor but still detectable in this region. So at the end, we have a protein which, despite a really low level of divergence at the primary amino acid sequence, has an overall organization, domain structure, and potential three-dimensional structure similar to the top6B subunit of Archaebacteria. Interestingly, in some species we could not find it, the whole protein. In particular, I'm indicating about in fungi, but fungi turn out to have proteins which we are confident are autologous, but only have the C-terminal region of the top6B, which is a transducer domain. And I will get back to that further in my talk. So one prediction, if our protein is actually a partner of SPO11, is that it should interact. Top6B is interacting with top6A. I showed you before the structure of the heterotetramer. This interaction is taking place at the C-terminal of top 6b in the transducer domain and at the N-terminal of the top 6a. And here you have alignment of the top 6a protein from Actibacteria with eukaryotic orthologs of SPO11. So there is really significant conservation, and the black dots indicate the region in the top 6a proteins from Actibacteria that are known to interact with the B subunit based on the crystal structure. Interestingly, in the mouse, so now I'm going to highlight the interaction by really testing specific interaction with the mouse proteins. The mouse has two splice variants of SPO11. And interestingly, they differ by this 38 amino acid insertion, one of the variants which is lacking this region, which is the potential region of interaction with the B subunit. So we predict that the beta, they are called beta and alpha, the beta form of mouse SPO11 has the putative domain of interaction with the B-like subunit. And in fact, hereby, this is a result of yeast-2 hybrid assays. So the first line is a full-length mouse top 6b-like protein, which does interact specifically with the SPO11 beta and not alpha. So that fits with what I just mentioned. And here we have, so far, not very precise, but a rough mapping of the domain of top 6BL, which is involved interaction, which is the C-terminal region transducer, also fitting with the prediction. More directly, we have now gained evidence of the interaction between SPO11 and the top 6B-like protein in the mouse by expressing in E. coli and doing various affinity purification. So these are the protein that when expressed in E. coli, and here they have specific tags, they were able to co-purify upon affinity purification. Here it's using a nickel affinity column, where when pulling down top 6BL protein allows to recover SPO11 beta in this case. So the protein form what could be a stable complex, and the evidence for the complex is also illustrated here by a gel filtration, where the two proteins actually co-migrate with, indeed, in this fraction in the gel filtration purification, indicating a stable complex for these two proteins. To evaluate the size, we currently have only a relatively preliminary idea about the size of the complex, and this is on glycerol gradient. Again, the two proteins co-purify, at least a fraction of them, at a molecular weight of the order of 200 kilodalton, which could be, but of course, much more needs to be done to know whether this complex is an heterotetramer. So that's about where we are in the really biochemical interaction and what we know about between these two proteins. The other very strong prediction, if this top 6B-like subunit is a direct partner of SPO11 functionally involved in the formation of break, is that the mutant should have a meiotic double-strand break phenotype. So we set to test this hypothesis, and we did that by generating a mutation in the gene coding for this protein in the mouse by CRISPR-Cas9, a null mutation disrupting the open reading frame. And in this mutant, we're going to test one of the easiest and relatively direct. I mean, there are some more direct tests, but to monitor double-strand break formation, we can use, as I presented in the introduction, the phosphorylation of H2AX as an indirect evidence for formation of breaks. And of course, we can look at the formation of double-strand break by detecting double-strand break repair foci. And there are various double-strand break repair protein that allows you to do that. And here is an example of RPA, which bind to the single-strand DNA, which are exposed upon double-strand break formation. So this is what you see in the wild-type cells, which these cells are, in this case, mouse spermatocytes at the beginning of meiotic prophase. Okay, and in the middle is another marker that allows you to label the axis of the chromosome. So the mutant essentially validates our hypothesis. There is a very strong reduction of H2AX, of gamma H2AX formation, no detectable RPA foci. So we infer that this is consistent with a defect in double-strand break formation. And there are other phenotypes that we analyze on these mice, both in male and female meiosis, and they all point to the same conclusion. So the conclusion of this part of this work is that we have very strong evidence that the breaks are generated by a complex involving these two proteins, the structure of which is modeled but remains to be really actually determined, and that's something we have to work on. As I mentioned, the GHKL domain is structurally conserved, but at the amino acid level still highly divergent, and it remains to be determined what is the activity which in topo6 protein is an ITP binding and ATP hydrolysis. And I'm going to highlight this by showing the implication on the mechanism. So the left side shows you the schematic of topoisomerase or topo6 reaction, DNA cleavage, well, ATP binding, dimerization, cleavage, strand passage, religation. Okay, so that's the step. So what's going on for our two partners? And now what I'm going to say is just a speculation and raising questions, because that's now where we are in this field to trying to understand. There are several options. It's not clear whether this B subunit, for the reason I just mentioned, is actually ATP binding. It may or may not, and that's going to be very important to determine, because ATP binding, whether it is actually part of the property of this complex, may also not only allow cleavage, but potentially a reverse reaction, which would be religation. That's another aspect, very exciting, is SPO11 is known as the break-induced activity, but since it evolved from a topoisomerase, maybe it's also able to religate, and that's a question. Now there are some other factors that may be involved, so maybe these two subunits, SPO11 and TOP6BL, don't have an activity by themselves. We know, and the genetic says that there are several other genes that are absolutely required for break formation, so they might be indirectly or directly involved. So the complex may actually be more involved additional partners. In addition, as I mentioned, that in fungi, some protein, in fact, the ortholog is REC102, is lacking the GHKL domain. We think the GHKL domain or something equivalent to the domain is actually carried by another protein, which is called REC104. But that leaves open a lot of divergence in how this GHKL domain and role may be actually coordinating the break activity. So there may be also variation in various species in how this break formation is regulated. And to me, it really opens many fascinating questions about the evolution of this, which is actually the evolution of sexual reproduction, because it's how meiosis arose during the evolution of eukaryotes is really a fascinating issue. As mentioned here, it appears that the activity, something conserved in topo6, has been evolved and to carry out break formation. But why not other topo2 isomerases have been recruited in other species? So that's a little puzzling to me. Of course, it's very remarkable that this very dangerous break induction that is taking place during meiosis involves a protein covalent intermediate. This is in principle, that's the way I see it, a safety belt to the break formation, because potentially you can reverse in case you don't have the homologue or an homologous chromatid to repair. So at some point during the evolution of this molecular machinery, this must have been very important. Then Topo 6 has an important another activity, which is decatenation. And in fact, biologically, it's known to be important in some organisms for endoreplication. So is there a link between a role of decatenation, of endoreplication, and evolution of meiosis? So now I'm just speculating and raising the proposition also that maybe in early meiosis involved first the transition from haploid to diploid, for which several possible ways have been, may have occurred. One is cell fusion, but another one is actually endoreplication. So I find actually the parallel very interesting. Okay, so I'm going to go to the second story. I'm just taking my time here, that I'm going to present. Second question, which is where does double-strand break occur? I take again this cartoon of break formation. On the left, just another representation of chromatin loops and chromosome axis to give you just a feeling of that there is a question of spatial organization, spatial control of these events. And here, I think a remarkable, an example of the remarkable work from Alex Jeffreys, which was one pioneering work in identifying recombination sites in the human genome. And this is just to illustrate that it has been known for many years that recombination occurs at specific sites in human or mammalian genome. And the question had been, what are these sites? And I'm going to, of course, make the story short to give you the conclusion, because that's what we discovered 6 years ago, and by the identification of this protein called PRDM9. So that's the finding that we made together with the lab of Peter Donnelly and Ken Pagan. So the way this protein works, as far as we can tell, I'm going to give you just a basic outline, and then you will see there are several questions, and where I'm going to give you some further hint on its mechanism of action. So this protein PRDM9 has two domains I'm going to talk about. One is methyltransferase, and one is DNA binding. So its key role in specifying the localization of meiotic double-strand break is the DNA binding domain, which is a tandem array of C2H2 zinc finger. And I'm going to tell you a little bit more about that. But just you can take the message, the conclusion that we came with from studies that I'm not going to present the result, just the conclusion. It has been published. PRDM9 binds through its zinc finger through specific sequences in the genome. There are multiple sites where PRDM9 can bind. It modifies histone, etric F4 trimethylation in particular on adjacent nucleosome. And then it recruits or activates SPO11 complex for break formation. Okay, so it's simple in a way. But of course complicated to figure out and to unravel all this. So for localization, key is the zinc finger. Zinc finger, a lot is known because many transcription factors have a zinc finger domain, and the C2H2 zinc fingers have been crystallized, the structure has been known, and a zinc finger, which is a 28 amino acid domain, has at least three major, it's not exclusive, but three major residues which have some coordinates, which are directly interacting with the DNA, in particular the base and the DNA helix. And these residues specify the DNA sequence specificity and the affinity. In other words, if you change these residues, you change DNA specificity. Okay, and that has been used in the zinc finger field also as a technology for zinc finger nuclease. So such a protein like PRDM9 has a tandem array, meaning that in fact you have, in this case, 12 modules that recognize each a 3 nucleotide sequence, and that's in tandem. So that's a 36 potential recognition sequence that is recognized by this tandem array. And this is an in silico predicted motif, which we know now that it's actually a very poor prediction because we have the in vivo motif, and it's only very partially similar to what can be predicted in silico. And the critical amino acids are this one, as I mentioned, right in the center of this alpha helix of the zinc finger. So from this work, in particular, the Camerini lab has really given a really genome-wide picture by developing a DMC1 chip technology, which allows you to map genome-wide by chip sequencing all the sites where double-strand breaks are formed, because double-strand breaks, after they are formed, ends are processed and DMC1 loads and the ends. So by retrieving DNA associated with DMC1, you map all the sites where the breaks are formed in the meiotic cells. And you can see these peaks are the sites where the breaks are formed. And in fact, all these peaks, I'm not showing you the data, have motifs which match predicted, or which partially match the predicted sequence recognized by the zinc finger of PRDM9. So here we are using this mouse with this PRDM9 allele, and we have a large number of sites. Now the thing is that we and others have discovered that there are different PRDM9 alleles that differ by the zinc finger domain. So this other mouse has a different tandem array. That's the only difference with the mouse to the left, is the zinc finger array of PRDM9. This different amino acid composition in the zinc finger array leads to a different affinity to the DNA with a different recognized motif or family of motifs. And in fact, when you do the DMC1 chip and you compare with these between these two mouse, there is no overlap between the sites where double-strand breaks are formed. Okay, and that was also one very important finding so in the field when we discover PRDM9, because it means that two different mice, as this one presented here, have a different genetic map. Then really the question and the result that we are currently working on, and I'm going to present briefly, is where does PRDM9 bind? Because the data I presented before where the breaks are formed. But we want to understand more precisely the relationship between PRDM9 binding to the DNA and break formation. And we set to do this, and the strategy was to take two different mice, and again using these different alleles of PRDM9 with different tandem array of zinc finger, so not the same from the previous slide, but just a different combination. The combination principle of the strategy is the same. And then on these different mice, we do not only the DMC1 chip sequencing, but also the PRDM9 chip sequencing. And as you will see, monitoring H3K4 trimethyl, because from data that I have not shown you, PRDM9 methylates the lysine 3 of H3K4. And as just to present you as it comes out from the browser, is that you can find highlighted here on the red rectangle some sites which you have a signal for PRDM9, H3K4 trimethyl, and DMC1. And this is specific to one mouse strain and absent in the other strain. Okay, so we have really a site that is recognized by this PRDM9 protein. Just a zoom of this. It's recognized by PRDM9, and at the same location in the genome, there is break formation and specific H3K4 trimethylation. Okay, so now we have a large number of sites in these two different mouse. I can discuss this at the end, but I'm not going to discuss that right now. The numbers are different, but in any case, because one of them has 10,000 PRDM9 sites, the other one 2,600. Of course, we have a knockout control, which is very important because we are using antibody, and then, well, that's one of the various controls absolutely required to eliminate non-specific signal, and we can eliminate some non-specific signal if needed from a small overlap between the two peaks, sets of peaks that are detected. So among these peaks, the majority, that is 73% of these PRDM9 peaks, actually overlap with DMC1. So that's what you expect. That's the model, so that's fine. All these sites that we call canonical sites have DMC1, have H3K4 trimethyl in a strain-specific fashion, of course, so we can distinguish these marks, which are deposited by PRDM9, from the H3K4 trimethylation that's deposited by other methyltransferase, and among which you find many of them at transcription promoters or enhancers, but these are different locations, except when a PRDM9 binding site ends up overlapping a transcription promoter, which by chance happens some of the time. We have also shown that there is H3K36 trimethylation, and so I can comment that on discussion if some people are interested. And these sites have a consensus motif for the PRDM9 zinc finger domain. So that fits the model, and everything is fine. So what was surprising is that we uncovered a class of sites, what we call class 2, which don't have DMC1. And then, of course, that was puzzling, and one of the first question is it relevant? Is it just DMC1 is not detected for some technical reason? We think this is very unlikely because the DMC1 chip is actually much more sensitive than the PRDM9 chip. But we reason we could test that by another mean, which is to examine the one property of recombination site, which is the increase of GC content due to GC biased gene conversion. I don't have too much time to explain the detail of this phenomenon that has been described and validated by several groups in the mammalian genomes, where it has been demonstrated that regions which are sites of recombination, you can look in humans, sites of recombination sites have an increased GC content and an increased propensity to fix GC substitution by a process for which the molecular mechanism is totally unclear, but it's somehow occurring during double-strand break repair on the process of gene conversion, and it could involve mismatch repair, or several possibilities are open. So that's another way to look at the trace of recombination. It's a historical trace of recombination that leads to this evolution towards the GC enrichment. And thus, we could look at these sites, the PRDM9, the canonical site, in fact, called class 1. You do see this blue line, which is making a peak here, is an indication of the increased fixation of substitution towards GC, precisely at the center of the peak that we know now are recombination site. Okay, so we have an evolutionary trace on the musculus genome of this recombination activity. And then we ask, what about these sites where we could not detect DMC1? If it's a technical artifact and if there is recombination activity, we should detect this effect. And in fact, we don't. So we really think that these sites are sites where PRDM9 binds, but there is no recombination activity. Okay, and then another very unexpected property of this class 2 site is the absence of any motif. If you take the motif recognized by the PRDM9 zinc fingers from one or the other mouse, the class 1 site has the motif right at the center of the peak determined by chip sequencing, and the other sites divided in A and B, but I go back to that in a second. The other side don't have any motif that looks like something that could be recognized by the zinc finger. So these class 2 sites have two unusual properties. They don't seem to be recognized by the zinc finger domain of PRGM9, and we don't detect recombination activity, but PRGM9 is going there somehow. And that's illustrated here, and they are actually, we subdivided the A and B sites, well, the class 2 sites in two subcategories, A and B. And what's the difference between the A and B subcategories is the presence of H3K4 trimethyl. So you see, so we have this different information, and if we use H3K4 trimethylation, we have a subcategory of class 2 sites where there is a very strong signal. In fact, these are promoters and enhancers, and they have all the other marks typical of promoters of enhancers. So PRGM9 is going for somehow to this promoter or enhancers region. This other category has no epigenetic mark whatsoever that we could put our hand on. This just really illustrates what I just mentioned is if you look this bar graph here, you have the class 2A sites which show a very high proportion of the peaks which overlap with transcription promoters and enhancers. The class 2B sites which don't have any specific chromatin signature we could identify turn out to have a motif. And this motif in the black 6 strain is a CTCF motif. So the CTCF protein could require a very long introduction because it's a complex protein involved in many processes, but I'm just going to summarize, tell you that it's an insulator protein, also interacting with coenzyme, regulating chromosome organization, and having multiple downstream consequences based on this function. So now that's, what I'm presenting you is really at the border of what we currently know, and we don't have really molecular direct information about this information, but that's what we're going to think about for trying to understand this observation. So what we did is that by immunostaining, we could detect the CTCF protein on chromosome axis. And we think this is a very exciting perspective. This is preliminary data in a way because this needs to be confirmed by various ways. But it leads us to propose that, in fact, these PRGM9 binding sites are indirect by interaction, presumably that could be mediated by interaction between PRGM9 and CTCF, which is located on the axis without involving, as I said, the zinc finger domain of PRGM9, and without generating break formation at these sites. But this loop to axis interaction in the field is something that is very important, or thought to be very important for the regulation of break formation. So we think we are capturing here an important event for the regulation of break formation. Remember that one particularity of these class 2 sites is, although they don't involve the zinc finger domain of PRGM9, they don't overlap between the two different strains we have been analyzed. So they are specific. So they have a localization specificity which is independent at their site from zinc finger. So we think that such interaction mediates some capture of other sites, but the localization of which is dictated by some constraint, meaning that depending where you bind by the zinc finger domain at the first place on the class 1 site, the other sites that are going to be interacted with occur at different positions. And just let me check my timing here. Okay. One important functional information we got by trying to understand this unusual PRGM9 binding site was by performing the same experiment in SPO11 mutant mice. Although we have a lower number of sites just because just the material, the chromatin we started with was lower abundance. What is very clear is that the class 1 sites, which are the one recognized by the PRDM9 finger, are SPO11 independent. So hopefully that's really what fits and is predicted by the model, because at least in a temporal order we imagine, because we have not proven the kinetics in mouse spermatocytes, but we predict that PRDM9 binds first and then will recruit SPO11. So these binding interactions are SPO11 independent. The class 2B sites, which are the what we propose being this loop to axis interaction or interaction with CTCF, at least for some of them, are also SPO11 independent, because that they are detected in this black 6 SPO11 mutant experiment. But interestingly, the class 2A sites, which are the interaction of PRDM9 with transcription promoters, are essentially lost. The molecular details now we have to figure out, and I can just propose or actually summarize the observation, because this is in particular with this interaction of PRDM9 to promoters, which here on the right panel, we have to conclude that somehow upon double-strand break formation, PRDM9 may be displaced or relocated to other sites, and for some reason captured by some other interaction to promoter region. There are various alternatives, and that's the model may change depending on the interaction. We're looking for interaction, but this has been relatively difficult to challenge. We think we have more, it would be right in the first place really important to expand further this interaction, which we think highlights the interaction between loops and a chromosome axis. I'm going to summarize these two molecular aspects. Now I think we can integrate many parts that we have been trying to build up over the years to understand the various levels of double-strand break formation and the regulation of this process. So, and this diagram highlights that in mammals, so mouse and humans at least, have this PRDM9 function. And I'm saying this because PRDM9 is not conserved in other animals, is not conserved in plants or fungi. So this is a mammalian, let's say for the moment, it's a mammalian specific function that allows to tether and to direct recombination activity to specific sites. So it's binding there. It probably recruits, so what I've shown you, the SPO11 top 6BL complex. This complex could be recruited at this stage or at the stage that we think is the following one, which would imply the contact or interaction of this PRDM9 interacting region with axis. So SPO11 could come in later or before, that's unknown. Of course, this model is assuming that there is an interaction between PRDM9 sites and axis, and we don't know how this is working. We think the H3K4 trimethyl is playing an important role, but for the moment we don't know the detail, and in fact we are mostly following hypotheses and the data that has been demonstrated in yeast about this interaction. The important thing is that double-strand break, we predict, should be formed on chromosome axis, and one support of that is that there are several proteins, and we have identified MEI4 and REC114. Attila Toth has identified IO1 recently, are located on the axis. So you ensure somehow that breaks are formed on the axis by having essential component for the activity specifically located on this part of the structural chromosome. So the two machinery, PRDM9 that I presented, of course, has many evolutionary implications. I didn't go into that, but I can discuss about this because it's mammalian specific and it has other implications on genome evolution. Of course, also the other thing just to remember also from this talk is the key role of this topoisomerase-like complex. And by like, there are many open divergence or similarities that we are trying to understand that is involved in the formation of DNA breaks. So I'd like to thank the people who did the work. The SPO11 top 6BL work was done with, of course, a great collaboration with Mathilde Grelon that is mentioned here, and with Henri-Marc Bourbon, who was fantastic in identifying really remote orthologs in all eukaryotes. Thomas Robert really drove the project really fantastically. He's a research associate in my lab, and with Alex Knorr, a very great PhD student also who did major experiment for this project. The PRDM9 binding site mapping has been very challenging and very difficult. We spent actually many years before to really get this result that I presented. And that's fantastic work by Corinne Gray and the bioinformatician Julie Clément. And I'd like to thank the funding agency, and I thank you for your attention.
Thank you, Bernard, for a wonderful talk, and we have time for questions. Okay.
So you mentioned a certain number of sites, class 2A sites, where SPO11 is found in the chromosome and is binding SPO11. So is it possible that the class 2A sites are in fact by default targeted by SPO11 that you see when PRDM9 is absent?
Yeah, I see what you mean. Yes, so we have looked, so the question is something really I didn't get into this other really highly related point, which is, I think it was on the slide, but I didn't comment on this. In the absence of PRDM9, the double-strand break machinery, including SPO11, goes to transcription promoters. Okay, and that has been shown by Dan Camerini Otero's lab. And so it's a break formation pathway, essentially, that is actually highly similar in the principle of what has been described in yeast and is probably occurring also in plants. So we can call it a default pathway. The answer is that the breaks that are formed in the absence of PRDM9 essentially go into any promoter, all promoters. Most of promoters are actually double-strand break sites in the absence of PRDM9. So yes, we do see an overlap between this PRDM9 binding site and promoters in general. And even if we look at the subset of promoters that are the default sites, we also see the overlap. So you see, yes, we see an overlap, but it doesn't tell us much more because apparently most, I mean not to say all, most promoters are used as a default in the absence of PRDM9 for break formation. And here we have a small number of promoters because the sensitivity of the chip is that's the way it is, and PRDM9 is going there. So I don't know if I really answered to your question, but you see there is nothing more specific we can test as far as I can tell.
Yes.
So in absence of topo-6B, still using the SPO11 there?
So you're asking if in the absence of topo-6B, like we see SPO11, you're asking if we have some immunolocalization or way to detect SPO11. We don't. Okay, so the answer is I cannot answer this question because we do not not able to detect SPO11 by immunofluorescence. So yes, essentially I don't have the answer.
You have Topo B. And why do you need Topo B? SPO11 itself not enough to make the breaks.
Oh, this is your question. So what we show is absolutely yes. SPO11 alone is unable to make a break. So that's a very clear conclusion. But for us, it really makes sense because based on the similarity I discussed with topoisomerase, you need the A and B interaction and organized in some way to make a break and yes.
So the difference between these two mouse strains is very striking. You have one with 10,000 hotspots and one with 2,000.
Mm-hmm.
So is there some compensation in the one with 2,000 to make the combination work normally? Are they all hotter?
Yeah. So okay. So that could be maybe a little long to explain the various simply. So there are two aspects on this observation. One is why there are different numbers, and then what are the consequences on break formation. So briefly, what I can answer, and we can discuss more, but is that on break formation, there is no difference. Okay, so you can reduce the number of detectable binding sites, which in terms of binding are not hotter for PRDM9. No, but I mean, I don't want to push this too much because making conclusion on strength of binding by a chipsec is really okay. It's in particular between two experiments, unless you have internal control for this. So this is really borderline of. But what we know is for sure is that the level of DMC1 is essentially the same. So maybe we could reduce PRDM9 binding, and that does not affect break formation. So this means that maybe, well, the way I see it is that for break formation, you need PRDM9 to bind, probably H3K4 trimethyl. And then you need a third step is to activate SPO11. But this is not just an enzymatic reaction. Okay, and then there is a difference in the number, and we think this is due to another feature that is very important for the evolution of PRDM9, which is, but you have, I'm sure you know that, it's a hotspot erosion. So we think in the strain that where this black 6, so this one is a black 6 PRDM9 allele, which is in a black 6 genome. So historically, this PRDM9 allele has been in the contact of the genome for, hundreds, thousands of generations. And because PRDM9 is actually eroding its own binding sites, and that has been shown in human and in mouse, the binding sites for PRDM9 tend to decrease progressively during evolution. And we think that this lower number is due to that, because the other one is coming from another mouse which is phylogenetically distant.
In the strain of 2000, do you start to get more of those promoter breaks? Is there any more breaks?
Yes, actually yes, but actually there is some data from Camerini lab, because they had done just the DMC1 chip, so they don't have the PRDM9 information, but if in their published paper, you can see that there is a higher, I don't know if it's significant, but there is a higher proportion of breaks that are formed in promoter region in this context. Yes, absolutely.
Bernard, I had one question that I was carrying with me for as long as the PRDM9 story came out. it has the zinc finger motifs, but they come in two groups. One is the tandem repeats, and then there's one zinc finger more towards the N-terminus, a singular zinc finger that seems to be apart from the repeats. Does that have any functional specialization in PRDM9, or is that just a remnant, or
They are
maybe signaling that it's not involved in DNA binding, but maybe involved in other interactions with small molecules, with proteins?
yeah, there is a zinc finger, but flanking the set domain.
Mm-hmm.
And there is another which has a helix which is called the zinc knuckle because it has a different organization. So these are structural components presumably important for the set domain for methyltransferase activity. Mm-hmm. We have done a structural study on the set domain. And yes, we think one is important really for the structure of this whole domain. And then there is another one which is isolated, and this one we don't have information. I don't expect they are involved in DNA binding, this other zinc finger. We're doing some in vitro DNA binding assay, and for that we take only the tandem array of zinc fingers. And well, we can recapitulate, but of course DNA binding and affinity. So no other indication, probably not likely to be involved in DNA binding.
Yes?
So have you tried chip for CTCF?
For which one?
CTCF.
CTCF. No, we have not tried chip for CTCF. There was on the slide a graph, but I went fast. There is a CTCF chip sequencing from ENCODE, so it has been done. So the CTCF bindings, so it has been done, but for us what we need is to have the information on these spermatocytes, and it has been done in other cell types. Well, it has been done in ES cells and other cells. The data that I presented, it has been done on mouse testis, which is a mixture of different cell types, so which is not what we would really need. So we need to have more spermatocytes, pure population, and that we have not done this experiment, not yet.
Steve?
So you mentioned there's a splice variant that's SPO11, the alpha, right?
Mm-hmm.
And it doesn't interact with top 6.
It does not, yeah.
It does not. So do you know whether it has a role? Do you know whether it's part of the whole strand break repair? So what I'm talking about,
Yeah.
I'm thinking that what you want is you want to make a break, but then you want to make the heterotetramer unstable. So do you know when you have a hetero-dimer of alpha and beta in the functional complexes? Or maybe that's biochemical?
No, okay, that specific experiment tried to build an heterodimer with SPO11 alpha and beta and to see if these two guys will dimerize. We have not done that.
So how about in your CRISPR knockout, can you complement, if it's a deletion, can you complement with this beta?
Yeah, so maybe what I can tell you is that some genetic had been done by, was in a work that had been done by Camerini and some by Maria Jezin and Scott Kinney. So that leads to the information is that SPO11 beta alone gives rise to almost complete fertility. But I say almost because meiosis in the presence of SPO11 beta alone shows some defect of let's say on recombination. It has been tested by synapsis, which is a proxy for recombination on the X and Y chromosome in the pseudo-autosomal region. So based on these two publications, the proposition is that SPO11 alpha is required for maybe double strand break formation. I'm making a little bit oversimplistic, but maybe for double strand break formation in the pseudo-autosomal region on the X and the Y. because we think it's not interacting with top 6BL, this role of SPO11 alpha could be, I don't know, to inhibit a repressor of for SPO11 beta to act or something funny like that. So yes, it's a funny, but it may be a way to regulate. Maybe there is still some things we don't know, and another way, it's still a way to turn off double-strand break formation by having, I mean, as you say, maybe SPO11 alpha coming into the heterotetramer, and then you make an inactive heterotetramer. Because SPO11, one very important regulation of SPO11 must be how you turn off this activity. And one of my ideas maybe is involved in turning off. Yeah.
Nev?
I guess one way to prevent religation of that SPO11 complex is to make the EPP release very slow. So have you guys looked at the EPP cycle in any way?
To make what very slow?
ADP release?
ADP, yes. Yes, I will, but really what I want to emphasize is that on this protein, this top 6B-like protein, we have no evidence for ATP binding yet, right? So, I mean, we are just spending, we need to spend more time to purify the protein and to do ATP binding assays. So for us, the first question is, does it bind ATP? So it has a fold, it has some motifs are absent. So, you know, it's an open question. It could or not bind ATP. And then, yes, if it binds, I think it will be important to look at ATP hydrolysis. And then when you hydrolyze ATP, the ATP, the phosphate release. I mean, the phosphate release is the key step in the dimerization. That we will be able to look at, but the very first step is whether it binds ATP. Is that what we have to answer?
One last question.
From an evolutionary perspective, do you think that the function of PRDM9 leads to avoid break-induced mutagenesis in the motors?
Well, break-induced mutagenesis. You know, one question, for instance, in the field of double-strand break repair, let's say I could put in general term, is there is DNA synthesis associated with double-strand break repair. And there has been, well, there is data and there is discussion whether this synthesis, this process of synthesis, which extend over relatively short, it's you know, several hundred nucleotides long, or assuming, well, the length is not really known, whether this DNA synthesis is mutagenic or not. So there are papers, there's data in yeast showing that, yes, it is mutagenic. And there is actually this very old genetic work showing that there is mutagenicity associated with double-strand break repair. So this, for me, this is the question. So changing where the double-strand break would occur may not change the mutagenicity problem unless, I don't know, unless you have specific ideas or suggestions that a repair in a promoter region should be more mutagenic. Well, I think there are many places where you want to avoid mutagenesis, and that's also, you know, in coding sequences. So. Well, actually, we know from the mapping of the breaks genome-wide that I think about 40% of the breaks are in genes. In genes, because which could be exon introns. So, yeah, I understand there is a question around this, right? But I don't think really there is a specific problem related with mutation in promoter region that is related to PRDM9 function. But yeah, it's still, we still have to evaluate. I mean, I must say this, why it's this very complicated and even system has evolved is, I think we don't know. So I agree that there is a question behind this.
All right. Let us thank Dr. DeMassey for an exciting seminar. Thank you. So we have an ensuing discussion with the speaker, with some enrolled students of the class, GG 295, and so we're going to stay here and just form a